somewhere right now, on a warm coral reef in the Indo-Pacific, a small fish is settling down to rest for the night. It has found a quiet patch of sandy bottom, tucked among the broken fingers of staghorn coral, and it does not notice the slight disturbance in the sand a few inches away. Something is rising from beneath the surface. Something slow, something patient. A fleshy tube, no thicker than a drinking straw, extends silently through the water toward the sleeping fish. At the end of that tube is a weapon so precise, so fast, and so chemically devastating that the fish will never feel what happens next. In less than 100 microseconds, a hollow, barbed tooth will fire from the tip of that tube with an acceleration rivaling a bullet leaving the barrel of a gun. Inside that tooth flows a cocktail of several hundred specialized toxins, each one engineered by millions of years of evolution to shut down a different part of the victim's nervous system simultaneously. Within 2 seconds, the fish will be paralyzed. Within a few more, it will be swallowed whole, consumed alive by a creature most people would dismiss as nothing more than a pretty seashell. This is the geography cone snail, Conus geographus. It measures about 4 to 6 inches long. It has no legs, no claws, no teeth in the traditional sense. It moves at a pace that would embarrass a garden slug, and yet, drop for drop, this animal produces one of the most complex and lethal venoms on the planet. A single geography cone carries enough poison to kill more than a dozen adult humans. There is no anti-venom. If you are stung, the only medical treatment available is to keep you breathing, keep your heart beating, and wait for the toxins to wear off. That is it. That is the state of modern medicine against this creature. Among divers and marine biologists who work in tropical waters, the geography cone has earned a grim nickname. They call it the cigarette snail, a piece of dark humor suggesting that if one of these animals stings you, you have just enough time to smoke one last cigarette before you die. The reality is somewhat less dramatic. Most fatal stings take between 1 and 5 hours to kill a healthy adult. But the nickname captures something important about the way people who spend time in these waters think about this animal. It is respected, it is feared, and it is never, ever handled carelessly. But here is what makes the geography cone snail truly extraordinary, and it is not the killing. It is the chemistry, because buried inside that venom, alongside the neurotoxins and the paralytic agents and the molecules that stop your heart, scientists have found compounds that may represent some of the most important pharmaceutical breakthroughs of the 21st century. Pain medications are 1,000 times more powerful than morphine, with none of morphine's addictive properties. A specialized form of insulin that acts faster than anything human bioengineers have managed to create. Molecules that target specific receptors in the human nervous system with a precision that synthetic drugs cannot match. The geography cone snail is simultaneously one of the deadliest creatures on Earth and one of the most promising sources of life-saving medicine ever discovered. And to understand how both of those things can be true at the same time, you need to understand how this remarkable animal hunts, how it evolved, and what exactly is inside that venom-tipped harpoon. So tonight, let us sink beneath the surface of a warm tropical sea. Let the current rock us gently and spend some time with the most dangerous snail alive. To appreciate what the geography cone snail is capable of, you first need to understand what you are looking at when you see one, because at first glance, it does not look dangerous at all. It looks like a seashell. A beautiful one, actually. The shell is broad, thin-walled, and cylindrical inflated, tapering to a low, flat spire at one end. Adult specimens range from about 1.7 inches to over 6 inches in length. The ground color is typically a pinkish white or cream, sometimes with a faint violet or reddish tint. And over that base color, the shell is painted with an intricate network of brown and chocolate mottling, arranged in loose bands that resemble the contour lines on a topographic map. That is where the animal gets its name. The pattern on its shell looks like the markings on an old geographical chart, and shell collectors have prized geography cones for centuries because of that distinctive, almost hand-drawn appearance. But the shell is just the house. The animal inside is where things get interesting. If you could gently turn the shell over and peer inside, you would see a remarkably complex organism packed into that smooth, conical fortress. The soft body of the geography cone fills the interior of the shell, anchored by a powerful columellar muscle that allows the snail to retract deep inside when threatened. The mantle, a thin sheet of tissue that lines the inner surface of the shell, is responsible for secreting the calcium carbonate that forms and repairs the shell throughout the snail's life. As the animal grows, it adds new material to the lip of the shell, gradually expanding its home. A geography cone does not change shells the way a hermit crab does. It builds one shell and lives in it for its entire life, adding to it year after year. This is one reason shell collectors find large specimens so appealing. A big geography cone represents years of slow, steady construction. Each layer of shell material, a record of growth. The shell itself, despite its beauty, serves a primarily defensive purpose. It is the snail's armor, its retreat, its last line of protection against predators. But the geography cone is more interesting for what it does outside the shell than for the shell itself. Like all cone snails, the geography cone is a gastropod mollusk. Its soft body is protected by that hard, calcareous shell, and it moves using a single muscular foot that extends from the shell's opening or aperture. Two small eyes sit on short stalks near the front of the body. But these eyes are relatively primitive. The geography cone does not rely on vision to find its food. Instead, it uses a structure called the siphon, an extension of the mantle tissue that functions like a chemical antenna. The siphon draws water over specialized sensory cells that can detect the faintest traces of chemical compounds dissolved in the surrounding water. This is chemoreception, the ability to taste the ocean around it, and it is how the geography cone locates prey in the dark. Because this animal is nocturnal, during the day, it buries itself in the sandy substrate of the reef, completely hidden from view. Its brownish shell, covered in a thin layer of protein-based material called the periostracum, blends perfectly with the surrounding sand and coral rubble. It waits there, motionless, sometimes for hours. Then, as darkness falls and the reef transforms from a sunlit wonderland into a shadowy hunting ground, the geography cone stirs. It begins to move slowly and deliberately across the sandy bottom, its siphon testing the water for the chemical signatures of sleeping fish. And it is what happens next that has captivated scientists for decades. The geography cone's weapon system is a biological marvel that has no real equivalent anywhere else in the animal kingdom. It consists of several integrated components, each one essential to the snail's ability to hunt creatures that are, by any reasonable measure, vastly faster and more agile than it is. The first component is the venom gland. This is a long, convoluted duct that runs much of the length of the snail's body, coiled like a compressed spring inside the shell. The cells lining this duct synthesize hundreds of different peptide toxins, each one a small chain of amino acids folded into a precise, three-dimensional shape. These peptides are collectively known as canotoxins, and they represent one of the most chemically diverse arsenals in nature. A single geography cone snail may produce between 100 and 200 distinct canotoxins in its venom at any given time, and each one targets a different molecular receptor in the nervous system of its prey. The second component is the venom bulb. This is a muscular sac connected to the venom duct. When the snail is ready to strike, the bulb contracts and forces venom from the duct through the weapon's delivery system. The third component is the radula sac. In most snails, the radula is a ribbon-like structure covered in tiny teeth, used to scrape algae off rocks. But in cone snails, the radula has been radically modified over millions of years of evolution. Instead of a ribbon of teeth, the cone snail's radula produces individual, hollow, harpoon-shaped teeth, each one about the length of a small sewing needle. These teeth are made of chitin, the same material found in crab shells and insect exoskeletons, and they are barbed along one edge like a fishhook. The radula sac stores about 20 of these teeth at any one time, each in a different stage of growth and development. Only one tooth is loaded and ready to fire. Once that tooth is used, it is discarded, swallowed along with the prey, and a new tooth is moved into position. The snail essentially manufactures its own disposable hypodermic needles. The fourth and final component is the proboscis. This is a long, flexible, muscular tube that can extend far out from the snail's body. Think of it as a hollow arm that the snail can aim and direct. The loaded harpoon tooth sits at the tip of the proboscis, connected to the venom duct, loaded with toxins and ready to fire. When all of these components work together, the result is one of the fastest and most efficient predatory strikes in the entire animal kingdom. For years, scientists suspected that cone snails were fast strikers, but no one knew exactly how fast. The problem was that the strike happened too quickly for conventional high-speed cameras to capture. Researchers at Occidental College in Los Angeles, working with collaborators at the University of California, Irvine, finally solved this problem by using ultra-high-speed videography, filming cone snail strikes at up to 58,000 frames per second. What they found was astonishing. The harpoon strike of the cone snail species Conus catus, a close relative of the geography cone, was measured at a peak acceleration exceeding 400,000 meters per second squared. To put that number in perspective, a car accelerating from 0 to 60 miles per hour generates an acceleration of about 3 to 4 meters per second squared. The cone snail's harpoon accelerates at roughly 100,000 times that rate. The entire strike, from the moment the harpoon begins to move to the moment it penetrates the prey, happens in less than 100 microseconds. That is 1/10,000th of a second. The researchers described the speed as comparable to a bullet being fired from a pistol. It puts the cone snail in the same category as trapjaw ants snapping their mandibles shut, and mantis shrimp smashing prey with their raptorial appendages, among the fastest biological movements ever recorded. The researchers had to film at 58,000 frames per second to fully capture the harpoon's movement. The lighting required was so intense that the scientists conducting the experiments had to wear sunglasses in the laboratory. Even at 8,000 frames per second, the initial filming speed, the harpoon moved too fast to see. It took months of refinement to achieve the right combination of frame rate, illumination, and subject positioning to finally capture the full strike sequence on camera. But how does a snail, of all things, generate that kind of speed? The answer lies in a mechanism that engineers call a latch-mediated spring system. The cone snail does not rely on muscle speed alone to launch its harpoon. Instead, it uses a process of energy storage and rapid release. Here is how it works. The muscular venom bulb contracts, building up hydrostatic pressure inside the proboscis. This pressure pushes against the base of the loaded harpoon tooth. But the tooth is held in place by a tight ring of muscular tissue at the tip of the proboscis, a biological latch that prevents the tooth from moving forward until the pressure behind it reaches a critical threshold. When that threshold is reached, the latch releases, and all of that stored energy discharges in a single, explosive burst. The pressurized fluid rockets into the bulbous base of the harpoon, launching it forward at extraordinary speed. The tooth punches through the prey's scales or skin. The barbed tip lodges in the tissue, and venom begins flowing through the hollow shaft of the tooth directly into the victim's body. The base of the harpoon never actually leaves the proboscis. It remains anchored there, tethered to the snail, so that after the prey is paralyzed, the snail can reel it in like a fish on a line. Researchers have described this as a taser and tether strategy. The venom acts as the taser, instantly incapacitating the prey, while the harpoon acts as the tether, keeping the prey connected to the snail so it can be swallowed. But the geography cone snail does not always hunt this way. It has a second, even more unsettling method of capturing prey, and this method is unique among cone snails. It is called net hunting. Instead of extending its proboscis and firing a single harpoon at a single fish, the geography cone slowly opens its mouth, which can stretch and distend to an almost unbelievable degree, expanding like a balloon or a parachute. The gape of its mouth, or rostrum, can stretch wide enough to engulf a fish several inches long. This is not the image most people have of a snail. This is a predator unfolding a living net. The snail extends this gaping maw toward a school of small fish resting on the reef at night. And before it fires a single harpoon, it releases a cloud of chemical compounds into the surrounding water. Picture it, the dark water of the reef at night, barely illuminated by starlight filtering through the surface, and a slow-moving snail exhaling a plume of invisible chemicals that drift through the water toward the resting fish. The chemicals wash over the fish, entering their bloodstream through the delicate, permeable tissues of their gills. And within moments, the fish becomes sluggish, disoriented, and unable to swim. Their fins droop. Their eyes glaze. They drift downward, barely twitching. Then, while the fish are still dazed and floating, the snail engulfs one or more of them in its distended mouth and fires the harpoon inside, finishing the job. Several hours later, the snail will regurgitate the fish's bones and scales, cleaning off every scrap of flesh, along with the spent harpoon tooth. That cloud of chemicals is where the story of the geography cone snail gets truly extraordinary. Scientists have given it a name. They call it the Nirvana Cabal. The Nirvana Cabal is a cocktail of bioactive molecules, and it represents one of the most sophisticated chemical weapons ever documented in the natural world. To understand why scientists find it so remarkable, you need to understand what is in it and what those ingredients do. The bulk of any cone snail's venom consists of canotoxins, small peptide molecules that interact with specific proteins in the nervous system. Different canotoxins target different proteins, and the specificity with which they do so is almost eerie. Some block voltage-gated sodium channels, preventing nerve cells from generating the electrical impulses they need to fire. When sodium channels are blocked, nerves go silent. Muscles stop receiving signals. The body's entire electrical communication network begins to shut down. Others block potassium channels, which are essential for repolarizing nerve cells after they fire, meaning the nerves cannot reset and fire again. Still others block calcium channels, which control the release of chemical neurotransmitters at synapses, the junctions where one nerve cell communicates with the next. When calcium channels are blocked, the synapses go silent, even if the nerve cells upstream are still functioning. And yet, another class of canotoxins interferes with acetylcholine receptors at neuromuscular junctions, the critical connection points where motor nerves signal muscles to contract. Some canotoxins do the opposite of blocking. Instead of shutting things down, they force ion channels to stay open, causing a massive, uncontrolled flood of electrical activity. Nerves fire continuously and uncontrollably. Muscles contract violently and cannot relax, and the body is overwhelmed by a storm of chaotic signals. This is called excitotoxic shock, and it is one of the first effects a fish experiences when stung by certain cone snail species. When multiple canotoxins hit these targets simultaneously, the effect on the prey is catastrophic. the nervous system essentially short-circuits. Muscles lock up. The heart may stop. Breathing ceases. The Filipino biochemist Baldomero Oliveira, who pioneered the study of Conus snail venom at the University of Utah, has described the effect of a geography Conus sting on a human as comparable to being bitten by a cobra while simultaneously eating fugu, the Japanese pufferfish known for its paralytic toxin. It is a vivid comparison because the geography Conus venom attacks the body on multiple fronts at once, using different chemical pathways to produce overlapping waves of paralysis, pain suppression, and cardiovascular collapse. But the Nirvana cabal contains something beyond conventional neurotoxins. In 2015, researchers discovered that the geography Conus snail produces a specialized form of insulin as a component of its venom. This was a first. No other animal on Earth had ever been documented using insulin as an offensive weapon. Insulin, as most people know, is the hormone that regulates blood sugar. When your pancreas releases insulin into your bloodstream, it signals your cells to absorb glucose, lowering your blood sugar levels. This is a normal and essential process, but if too much insulin enters the bloodstream too quickly, blood sugar plummets to dangerously low levels, a condition called hypoglycemic shock. The victim becomes confused, disoriented, loses coordination, and eventually loses consciousness. The geography Conus snail has evolved a weaponized version of insulin, chemically distinct from its own metabolic insulin, and remarkably similar in structure to the insulin found in fish. This weaponized insulin is released into the water as part of the Nirvana cabal. When it washes over a school of fish and enters their bloodstream through their gills, their blood sugar crashes. The fish becomes sluggish and disoriented, essentially sedated by a massive, involuntary insulin overdose. The snail then moves in to feed. But the weaponized insulin is only half the story. In 2024, researchers at the University of Copenhagen made another startling discovery. The Nirvana cabal also contains a molecule that mimics somatostatin, a hormone produced by the pancreas in vertebrates. In healthy animals, somatostatin helps regulate the release of other hormones, including glucagon. Glucagon is the hormone that counteracts insulin. When blood sugar drops too low, the pancreas releases glucagon, which tells the liver to release stored glucose back into the bloodstream, bringing blood sugar back up by releasing a somatostatin mimic along with the weaponized insulin. The geography Conus snail blocks the fish's ability to fight back against the insulin assault. The fish cannot release glucagon. It cannot raise its blood sugar. The insulin drives blood glucose down, and the somatostatin mimic prevents the body from correcting the problem. The result is prolonged, inescapable hypoglycemic shock. The fish drifts in a stupor, unable to flee, unable to resist, while the snail closes in. The researchers described this as combinatorial chemical mimicry. The geography Conus snail has independently evolved molecules that imitate two distinct vertebrate hormones and deployed them together as a coordinated biochemical attack on the prey's metabolism. It is a strategy that has no known parallel anywhere else in nature. And here is one more detail that makes the venom of the geography cone snail so remarkable. Only about 15 to 20 of the 100 to 200 canotoxins in the venom are actually used for hunting. The rest appear to be used for defense against predators or for competitive interactions with other cone snails, or for purposes that scientists have not yet identified. Each individual canotoxin is produced by a different gene, and these genes are among the fastest evolving in the animal kingdom. The venom of a geography cone snail collected in the Philippines may contain a subtly different cocktail of peptides than one collected in Australia, because the genes encoding these toxins are constantly mutating, diversifying, and adapting to local conditions and local prey. It has been estimated that across all 700 or so species of cone snails, there may be more than 50,000 distinct canotoxin compounds waiting to be discovered and characterized. That is not just a venom, that is a library. To understand how a snail ended up with a ballistic harpoon and a pharmaceutical-grade chemical arsenal, you have to go back a long way. A very, very long way. The story of mollusks, the phylum to which cone snails belong, begins in the early Cambrian period, roughly 540 million years ago. In 2024, paleontologists from the University of Oxford described a remarkable fossil from Yunnan province in southern China. The creature, named Shishania aculeata, was a small, flat, shellless slug covered in tiny spiny cones made of chitin. It had a muscular foot on its underside, much like a modern slug, and it crept along the ancient seafloor approximately 514 million years ago. Shishania represents one of the oldest known mollusks, and it tells us something important about where the entire group started. The very first mollusks were not elegant, coiled creatures with beautiful shells. They were armored, spiny, slug-like animals, inching across the bottom of a primordial ocean that was almost unrecognizably different from the oceans we know today. From those humble beginnings, mollusks diversified with extraordinary speed during the Cambrian explosion, the great eruption of animal body plans that occurred between about 541 and roughly 500 million years ago. The major groups of mollusks, including the gastropods, which would eventually give rise to snails, the bivalves, and the cephalopods, all appeared during this interval. Early gastropods were exclusively marine, and their fossil record extends back to the Lower Cambrian, making them among the oldest known mollusk fossils. Over the next several hundred million years, gastropods colonized nearly every habitat on Earth. They moved into fresh water. They moved onto land. They developed lungs. They developed eyes on stalks. They diversified into more than 50,000 living species, making them one of the most successful animal groups on the planet, second only to insects in species richness. But the cone snails are a relatively recent branch on this ancient tree. The oldest known fossils of the family Canidae date to the Lower Eocene epoch, about 55 million years ago. During the Eocene, the world was warm. Sea levels were high, and vast tropical and subtropical oceans covered much of the globe. The ancestral home of cone snails appears to have been the Tethys Sea region, a huge body of warm water that once separated the northern and southern supercontinents. This was a world without ice caps, a world where tropical forests grew at latitudes that are now temperate, and warm, shallow seas teemed with life. It was the perfect incubator for a group of predatory snails that would go on to become one of the most diverse marine genera on Earth. From the Tethys, cone snails spread throughout the tropics, eventually colonizing the Indo-Pacific, the Caribbean, the Eastern Pacific, and the coasts of West Africa. Molecular studies suggest that all living cone snails belong to one of two major clades that diverged roughly 33 million years ago. Around the time the global climate began to cool and the Tethys Sea began to close, the diversification of cone snails since their origin has been extraordinary. They hold the record for the highest rate of diversification of any marine gastropod or bivalve group. New species are still being described every year, and the total number of recognized living species now exceeds 800. What drove this explosive radiation? Many researchers believe the answer lies in the venom, because each cone snail species produces a unique combination of canotoxin peptides. And because those peptides are so precisely targeted to specific prey species, the evolution of new venom combinations may have allowed new cone snail species to exploit new ecological niches without competing directly with existing species. In other words, venom diversity may have fueled species diversity, allowing dozens of cone snail species to coexist on a single reef by partitioning the available prey among themselves. The early cone snails were worm hunters. Molecular phylogenetic studies analyses that trace evolutionary relationships by comparing DNA sequences strongly suggest that the ancestor of all cone snails was a vermivorous species, feeding on polychaete worms in the sediment of warm, shallow seas. Predation on worms is the ancestral diet, and the majority of the more than 800 living species of cone snails still hunt worms today. These worm-hunting species tend to have slower-acting venoms and less elaborate delivery systems, because a polychaete worm buried in sand cannot exactly sprint away from danger. The venoms of worm-hunting cones are correspondingly less dangerous to humans. A sting from most vermivorous species is comparable to a bee sting. Painful, but not life-threatening. But at some point, certain lineages of cone snails made a dramatic dietary shift. Some began hunting other mollusks, targeting fellow gastropods, and even other cone snail species. These mollusk-hunting cones evolved thicker, more robust harpoon teeth capable of penetrating the shells of their prey, and their venoms shifted to target the unique physiology of other mollusks. A few of these species, particularly the textile cone, Conus textile, have also been implicated in serious human envenomations, though confirmed fatalities are extremely rare. And then a smaller number of lineages, perhaps 5 or 6 distinct groups, evolved the ability to hunt fish. This was a radical transformation. Catching a worm that lives in a burrow in the sand is a fundamentally different challenge from catching a fish that can dart away in a fraction of a second. The shift to fish hunting required faster-acting venoms, more powerful delivery systems, and entirely new hunting strategies. The geography cone snail belongs to one of these fish hunting lineages, a group called the Gastridium clade. This clade includes the geography cone and the tulip cone, Conus chalipa, both of which use the net hunting strategy and the nirvana cabal. The evolution of fish hunting in cone snails appears to have occurred independently at least twice, in different lineages, each time driven by the same fundamental pressure. A slow-moving predator that wants to catch fast-moving prey must evolve exceptionally potent and fast-acting weapons. And that is exactly what cone snails did. The rate at which cone snails have diversified is remarkable. They exhibit the highest rate of diversification of any marine gastropod or bivalve group. On a single coral reef in Papua New Guinea, researchers have documented as many as 36 different species of cone snails living side by side, each one specializing in different prey and deploying a different suite of venom compounds. This kind of species richness on a single reef is extraordinary, and it is driven by the incredible chemical diversity of cone snail venoms. Because each species produces a unique cocktail of canotoxins, fine-tuned to its specific prey, multiple species can coexist on the same reef without directly competing for the same food resources. The geography cone snail is a creature of the tropical Indo-Pacific. Its range extends across a vast swath of warm ocean, from the Red Sea through the Indian Ocean, across Southeast Asia, and south to the northern shores of Australia. From Exmouth in Western Australia, around through Darwin in the Northern Territory, to the coast of Queensland. Rare sightings have also been documented in New Caledonia, in the southwestern Pacific. Wherever you find healthy, shallow coral reefs with warm water and sandy bottoms, there is a reasonable chance that geography cones are somewhere nearby, buried just beneath the surface, waiting for nightfall. Imagine the coral reef at dusk. The colors are beginning to fade as the sunlight dims, the brilliant blues and greens of the shallow water deepening into inky purples and blacks. Parrotfish are settling into crevices for the night, secreting mucus sleeping bags around themselves. Wrasses are burrowing into the sand. Damselfish are tucking into their territories among the branching corals. The entire daytime shift of reef life is winding down, and the nighttime shift is gearing up. Moray eels are peering from their holes. Octopuses are beginning to prowl, and across the sandy patches between coral heads, shapes are beginning to emerge from the substrate. Geography cones, rising from their daytime hiding places, unfurl their siphons and begin their nightly hunts. Their preferred habitat is the sublittoral zone, the region of the seafloor that is always submerged, typically at depths from the low tide line down to about 200 meters, though they are most commonly found in much shallower water, on or near living or fragmented coral reefs. They favor areas where the substrate is a mix of sand, coral rubble, and reef debris, places where they can bury themselves during the day and emerge to hunt at night. They are less common on solid reef walls or in deeper water. The geography cone is strictly piscivorous as an adult. It eats fish. The size of the prey depends on the size of the snail, and it is not limited to any particular species of fish. Small reef fish in the 3 to 5 centimeter range are typical prey for smaller snails, while larger specimens can take fish up to 13 centimeters long. Interestingly, the geography cone has been documented eating venomous weaver fish, which possess their own defensive toxins. The weaver fish's defenses, however, are no match for the cone snail's overwhelming chemical assault. Reproductive behavior in the geography cone has been studied primarily in aquarium settings, and very little is known about courtship or mating in the wild. Some researchers have speculated that male geography cones may exhibit territorial behavior to secure access to mates, but direct observations of competition or courtship rituals in their natural habitat are lacking. What researchers have observed is that during copulation, the male crawls onto the female using its muscular foot and inserts a ribbon-like reproductive organ called the verge, analogous to a penis, into the female's genital opening near the anal notch. This position is maintained for at least 15 minutes before the male withdraws. 2 to 3 days after mating, the female lays several capsules of eggs on a hard surface, such as dead coral, rocks, or the underside of reef ledges. Each capsule contains roughly 40 eggs. The eggs incubate for 10 to 20 days before the larvae emerge. Geographic cone snails breed once a year, typically between April and September when the waters are warm. Sexual maturity may be reached between 6 and 12 months of age. A single breeding event can produce between 1,000 and 5,000 offspring, though the vast majority of these will not survive to adulthood. The larvae of cone snails go through a planktonic stage, drifting in the open water as tiny, free-swimming villagers before settling onto the reef and beginning their lives as bottom-dwelling predators. During this planktonic phase, the larvae are extraordinarily vulnerable. They are eaten by fish, by jellyfish, by filter-feeding organisms, by virtually anything that consumes plankton. The attrition rate is staggering. Of the thousands of larvae produced by a single mating pair, only a handful will survive long enough to settle onto the reef, find suitable habitat, and begin growing into juvenile snails. Very little is known about what happens during the transition from larva to juvenile in the geography cone. The early life stages of this species remain one of the least studied and most mysterious aspects of its biology. As adults, geography cones face a relatively short list of predators. Sea turtles, particularly hawksbill turtles, are known to eat mollusks, including cone snails, and their thick, keratinous beaks may provide some protection against the snail's sting. Rays can crush cone snail shells with their powerful, flat teeth. Certain species of fish, including pufferfish, triggerfish, and wrasses, may also prey on smaller cone snails. And then there are human shell collectors who have been picking up geography cones for centuries, drawn by the beautiful mottled patterns on the shell, sometimes with fatal consequences. During the day, the geography cone is essentially invisible. It lies buried in the sand, its shell concealed by a layer of periostracum and sediment. Only its siphon protruding slightly above the surface to test the water for chemical signals. It is inactive, conserving energy, waiting. Then, as the reef darkens and the daytime fish retreat to their sleeping spots, the geography cone begins to move. It crawls slowly across the sandy bottom, its siphon extended, following chemical trails in the water. When it detects a sleeping fish, it begins its approach. Slowly, deliberately, it moves closer. The proboscis extends, the harpoon loads, and then, in a fraction of a second, the strike comes. After feeding, the geography cone will regurgitate the indigestible parts of its meal, the fish's bones, scales, and spines, along with the used harpoon tooth. A new tooth is already being prepared in the radula sac, ready for the next hunt. In the wild, the geography cone's lifespan is estimated at 10 to perhaps 20 years, though reliable data on longevity in natural conditions is scarce. The first documented account of a human being stung by a cone snail dates to 1670, recorded in the Dutch East Indies, but it was a case in the Seychelles Islands in 1932 that first drew serious scientific attention to cone, snail, and venomation. A man was holding a geography cone in his hand, scraping the shell with a knife, presumably to clean it, when the animal stung him. Within an hour, his entire body was numb. 9 hours later, he still could not stand. The physician who treated him, a man named Hermit, was so fascinated that a creature smaller than his fist could completely incapacitate a healthy adult that he dissected the snail and discovered the radular tooth protruding from the proboscis. The venom duct coiled inside the body. 3 years later, in 1935, the first well-documented fatal case occurred on Hayman Island on the Great Barrier Reef. A man named Charles Garbut was collecting shells when he picked up a geography cone and put it in his pocket. The animal stung him through his clothing. He was dead within 5 hours. That specimen, the shell responsible for Australia's only confirmed cone snail fatality, is now on display at the Queensland Museum. The case drew widespread media coverage in Australia and is credited with helping to educate the public about the dangers of handling live cone snails. In the same year, another fatal sting occurred in Okinawa, with the victim dying within 3 or 4 hours. The snail in that case was considerably larger than the one that killed Garbut. And toxicologists have since noted that stings from larger geography cones tend to be more dangerous, regardless of the victim's age or health, simply because a bigger snail can inject a larger dose of venom. In 1964, a geography cone sting proved fatal in just 2 hours, one of the fastest fatalities on record. In each of these cases, the story followed the same tragic pattern. Someone saw a beautiful shell, picked it up, and was stung. In one particularly heartbreaking case, a young man picked up a geography cone shell to give to his girlfriend as a gift. He placed it on his shoulder to show his friends. The animal stung him through his shirt, and he died shortly afterward. The geography cone does not sting out of malice. It stings because it is defending itself. It cannot see or understand the creature holding it. All it knows is that it has been grabbed, squeezed, and exposed. And its only defense is the same harpoon it uses to hunt fish. Over the past 350 years, a database maintained by the researcher Alan Cohn has documented 141 confirmed cases of human envenomation by cone snails, of which 36 were fatal. The vast majority of those fatalities, 31 out of 36, were caused by a single species, Conus geographus. No other cone snail species has been conclusively proven to have killed a human being on more than isolated, unconfirmed occasions. The geography cone is dangerous for several reasons. First, it is the largest piscivorous cone snail species, meaning it can inject a larger volume of venom than its smaller fish-hunting relatives. Second, its venom has evolved to target the nervous systems of vertebrates, including fish, and humans are vertebrates. The canotoxins that evolved to paralyze fish prey work on human nerves and muscles as well. Third, the geography cone is unusually aggressive compared to other cone snails. Most cone snails will retract deep into their shell when handled. The geography cone will readily extend its proboscis and sting in self-defense. The symptoms of a geography cone sting follow a grim progression. The initial sensation is a sharp prick, sometimes described as similar to a bee sting, sometimes much more painful. Within minutes, the area around the wound begins to go numb. The numbness spreads rapidly, extending to the entire limb, then to the torso. Dizziness sets in. Speech becomes slurred. Vision blurs. The victim may experience nausea and difficulty swallowing. As the venom continues to spread through the body, voluntary muscle control deteriorates. The chest muscles that control breathing weaken. In severe cases, respiratory paralysis occurs, and without artificial respiration, the victim suffocates. Death, when it occurs, is typically caused by respiratory failure or cardiac arrest. One of the most unsettling aspects of cone snail envenomation is that the venom contains its own analgesic. It suppresses pain. This means that some victims may not immediately realize how serious their condition is, because the sting does not hurt as much as they would expect. By the time the neurological symptoms become apparent, the venom has already had time to spread throughout the body. There is no antivenom for cone snail stings. This is not due to a lack of trying. The problem is the sheer complexity of the venom. Each geography cone snail produces hundreds of different peptide toxins, and the specific combination varies between individuals and between populations. An antivenom would need to neutralize not one toxin, but dozens or hundreds of them simultaneously. Researchers have estimated that across all cone snail species, there may be more than 100,000 different bioactive compounds in their collective venoms. Developing a universal antivenom for that level of chemical diversity is, for now, beyond the reach of pharmaceutical science. The recommended first aid for a cone snail sting is straightforward but critical. Apply firm pressure to the wound using a pressure immobilization bandage if available. Keep the victim still and calm. Do not attempt to suck out the venom or cut the wound. Get the victim to a hospital as quickly as possible. If breathing stops, begin artificial respiration immediately. In a hospital setting, the primary treatment is supportive care, mechanical ventilation to keep the patient breathing, and cardiovascular monitoring to manage any cardiac effects. The goal is simply to keep the patient alive until the toxins are metabolized and cleared from the body. It is worth noting that despite the geography cone's fearsome reputation, fatal stings are exceptionally rare. 36 confirmed deaths in 350 years is a tiny number compared to the thousands of people who die annually from bee stings, snake bites, or jellyfish encounters. The geography cone is dangerous, yes, but it is not aggressive toward humans by nature. Nearly all human stings occur because someone picked up a live cone snail, either deliberately out of curiosity or a desire to collect the shell, or accidentally by stepping on one in shallow water. The simple rule for staying safe around cone snails is to never pick up a live one. Admire the shells from a distance. Leave them on the reef where they belong. In the early 1970s, a young Filipino chemist named Baldomero Olivera made a decision that his colleagues considered, at best, unconventional, and at worst, career suicide. After earning his doctorate at the California Institute of Technology and completing postdoctoral work at Stanford, Olivera returned to the Philippines to take a position in the Department of Biochemistry at the University of the Philippines College of Medicine. The laboratory he was given had almost no equipment. He could not possibly compete with well-funded American labs in his original field of DNA enzymology, so Olivera looked for a project he could pursue without sophisticated equipment. And he remembered something from his childhood. Growing up in the Philippines, a country of more than 7,000 islands, Olivera had been an avid shell collector. One of his high school teachers had encouraged him to take a more scientific approach to the hobby. And from a young age, Olivera had been fascinated by cone snails in particular. He even wanted to study them for his undergraduate thesis at the University of the Philippines. But his advisory committee talked him out of it, telling him he would not be able to accomplish anything meaningful with such an obscure subject. He graduated summa cum laude and valedictorian of his class in 1960, studied DNA biophysical chemistry at Caltech, and completed postdoctoral work at Stanford. But those cone shells never left his mind. Now, back in the Philippines with an empty lab and a stalled career, Olivera returned to his childhood fascination. He knew that cone snails were venomous. He knew that some of them could kill people. He knew the geography cone was particularly dangerous. And he reasoned that if he could extract the venom and purify its components, he would not need expensive equipment to do so. He could inject crude venom into mice and observe the effects, then gradually isolate the active compounds using basic biochemical techniques. He teamed up with a colleague at the university, Lord Cruz, a brilliant biochemist who would go on to become one of the most important figures in canotoxin research. Together, they began visiting the shell dealers at the markets during their lunch breaks, buying live cone snails, and building relationships with collectors and fishermen who could supply specimens. It was, by Olivera's own admission, a side project. Nobody thought studying snail venom would lead anywhere important. His grant applications were rejected repeatedly. Reviewers were skeptical that anything useful could come from studying the venom of an obscure marine snail. That side project changed neuroscience over the next several decades, working with collaborators including Lourdes Cruz in the Philippines and a succession of brilliant students at the University of Utah, where he eventually settled. Olivera and his team isolated and characterized dozens of individual conotixins. They discovered that these peptides were extraordinarily specific in their molecular targets. Individual conotixins would bind to one particular subtype of ion channel or receptor and ignore all others. This kind of specificity was exactly what neuroscientists needed. For years, researchers had been struggling to understand the individual roles of the many different types of ion channels and receptors in the nervous system. But they lacked the tools to selectively target one type without affecting the others. Conotixins provided those tools. Each one was like a molecular key that fit only one lock. One of the most important discoveries to come out of Olivera's lab was a peptide called omega-conotixin MVIA, isolated from the venom of Conus magus, the magician's cone. This peptide specifically blocks a type of calcium channel called the N-type, voltage-sensitive calcium channel, which plays a critical role in transmitting pain signals in the spinal cord. When this channel is blocked, pain signals cannot travel from the body to the brain. The effect is profound analgesia, pain relief, without the sedation, respiratory depression, or addictive properties associated with opioid drugs like morphine. This discovery led directly to the development of ziconotide, a synthetic version of omega-conotixin MVIA that is chemically identical to the natural peptide produced by the magician's cone. After years of clinical trials conducted by the biotechnology company Nurex, which was later acquired by Elan Pharmaceuticals for $760 million, largely on the strength of this one compound. Ziconotide was approved by the United States Food and Drug Administration in December of 2004. It was marketed under the brand name Prialt, a name that stands for primary alternative to morphine. Prialt must be delivered directly into the spinal fluid through an implanted pump. Because the peptide cannot cross the blood-brain barrier when taken orally or intravenously, this makes it expensive and invasive, limiting its use to patients with severe chronic pain who have not responded to other treatments. The pump is surgically implanted in the patient's flank or buttock, and a catheter tunnels beneath the skin to deliver the drug directly into the cerebrospinal fluid surrounding the spinal cord. Patients return every 40 to 60 days to have the pump's reservoir refilled. It is not a casual treatment. It is reserved for people whose pain is so severe and so resistant to other medications that this level of intervention is warranted. But for those patients, Prialt can provide extraordinary relief. And unlike opioids, there is no evidence that patients develop tolerance to ziconotide, even after years of continuous treatment. This is a critical distinction, one of the defining problems with opioid pain medications. is that patients gradually require higher and higher doses to achieve the same level of pain relief, a cycle that drives addiction and dramatically increases the risk of overdose. Ziconotide does not appear to trigger this cycle. The dose that works on day 1 continues to work on day 1000. In a world ravaged by opioid addiction and overdose deaths, a powerful non-addictive pain medication derived from snail venom represents a remarkable breakthrough. Perhaps the most extraordinary detail about ziconotide is this. During its development, the pharmaceutical company Elan tested every possible variation of the molecule, systematically mutating every amino acid to see if any modification would improve the drug's performance. In the end, they did not change a single atom. The synthetic drug that is sold as Priald is chemically identical, amino acid for amino acid, to the natural peptide that the magician's cone snail has been producing in its venom duct for millions of years. Evolution, it turns out, had already optimized the molecule. Human chemistry could not improve upon it. But ziconotide is just the beginning. The geography cone snail itself has contributed several promising compounds to the pharmaceutical pipeline. One family of peptides found in the geography cone's venom, called kinetokins, is a potent blocker of a type of brain receptor called the NMDA receptor, which is involved in pain processing, learning, memory, and the symptoms of drug withdrawal. Researchers are investigating kinetokins as potential treatments for epilepsy, neuropathic pain, and even as protective agents against brain damage after strokes. And then there is the weaponized insulin. When researchers at the University of Utah discovered in 2015 that the geography cone snail uses a specialized insulin to induce hypoglycemic shock in fish, the diabetes research community took immediate notice. The cone snail's venom insulin, called Conus, is the smallest functional insulin molecule ever discovered from any source. It is structurally distinct from human insulin in ways that are potentially very useful for drug design. Specifically, it lacks a portion of the molecule called the B-chain C-terminus, which in human insulin causes the molecules to clump together into hexamers, clusters of 6 insulin molecules that must dissolve before the insulin can become active. This clumping is the reason that even the fastest-acting commercial insulin formulations take 15 to 90 minutes to begin working after injection. The cone snail's insulin does not clump. It is immediately active. This has made it a subject of intense interest for researchers developing faster-acting insulin for use in artificial pancreas devices and for emergency treatment of diabetic crises. Studies in zebrafish and mice have confirmed that venom insulins from cone snails can significantly lower blood glucose in diabetic animal models. And multiple research groups around the world are now working to adapt these molecules for human therapeutic use. The cone snail's insulin is not yet ready for human patients, but Helena Safevi, a biomedical researcher at the University of Copenhagen, who has been studying cone snail venom chemistry for over a decade, has described the potential in straightforward terms. Studying cone snails gives researchers a starting point, a molecule that evolution has already spent millions of years optimizing. Rather than designing a drug from scratch, scientists can begin with a compound that nature has already tested and refined. Across all species of cone snails, an estimated 70,000 or more distinct conotixin compounds may exist, each one potentially useful as a research tool, a drug lead, or a window into the molecular workings of the nervous system. As of the mid-2020s, only a tiny fraction of these compounds have been characterized. The cone snail venom library is vast, largely unexplored, and almost certainly contains therapeutic molecules that no one has discovered yet. The irony is hard to miss. The most dangerous snail on Earth may also be one of the most valuable animals in modern medicine. So what does the future hold for the geography cone snail? In one sense, the news is cautiously good. Conus geographus is widely distributed across the Indo-Pacific. It is not currently listed as vulnerable, threatened, or endangered by the International Union for Conservation of Nature. Its population numbers are not precisely known, but its broad geographic range suggests that the species as a whole is not in immediate danger of extinction. But the broader picture for cone snails is more complicated. In 2013, researchers conducted the first comprehensive conservation assessment of all 632 recognized cone snail species for the IUCN Red List. They found that while roughly 3/4 of species were not considered at immediate risk, about 6.5% were classified as threatened with extinction, and another 4% as near threatened. 3 species, all endemic to Cape Verde off the west coast of Africa, were classified as critically endangered. Nearly 14% of species were classified as data deficient, meaning scientists did not have enough information to assess their status one way or another. The threats facing cone snails are the same threats facing marine life across the tropics. Coral reef degradation from ocean warming, acidification, and bleaching events is destroying the habitats that cone snails depend on. Coastal development is burying intertidal zones under concrete and sand. Pollution from agriculture and industry is degrading water quality. And the global shell trade, while less intense than it was decades ago, still removes living cone snails from reefs for sale to collectors. For the geography cone specifically, the decline of coral reefs is the most serious long-term concern. These animals are specialists of the coral reef environment. They depend on healthy reefs for shelter, for hunting grounds, and for the ecological communities that support their prey. As reefs bleach and die, the complex mosaic of microhabitats that supports dozens of coexisting cone snail species on a single reef begins to simplify and degrade. Fish populations shift. Prey availability changes. The intricate ecological relationships that have sustained cone snail diversity for 55 million years begin to unravel. And there is a deeper concern that goes beyond the survival of any single species. Every cone snail species that goes extinct takes with it a unique library of conotixin compounds. compounds that may never be characterized, never tested, and never developed into drugs that could save human lives. A 2003 paper in the journal Science warned explicitly about this possibility, arguing that the loss of cone snail species represents not just an ecological tragedy, but a medical one. Each extinction deletes an irreplaceable chapter from the natural pharmacopoeia. Ocean acidification poses a particularly insidious threat. As carbon dioxide dissolves in seawater, the ocean becomes more acidic, and this increased acidity makes it harder for shell-building organisms, including cone snails, to form and maintain their calcium carbonate shells. If acidification continues on its current trajectory, the shells of marine mollusks could literally begin to dissolve by the end of this century. For a cone snail, the shell is everything. It is protection against predators. It is a shelter from environmental stress. It is the foundation of the animal's body plan. A world in which cone snails cannot build strong shells is a world in which cone snails cannot survive. There is also the matter of the shell trade. For centuries, cone snail shells have been prized by collectors for their intricate patterns and colors. The geography cone, with its distinctive map-like markings, has always been among the most sought-after species, while the global shell trade is somewhat more regulated now than it was in previous decades. Live collection of cone snails continues in many parts of the Indo-Pacific, and the cumulative effect of removing breeding adults from reef populations is difficult to assess. In some areas, localized overharvesting may be quietly reducing populations of species that no one is monitoring. The connection between cone snails and human medicine adds another layer of urgency to their conservation. Every species of cone snail produces a unique suite of canotoxin peptides. With more than 800 known species and potentially 50,000 or more distinct venom compounds across the genus, the collective venom library of cone snails represents one of the largest untapped reservoirs of bioactive molecules on the planet. Only a tiny fraction of these compounds have been characterized. The vast majority have never been tested for pharmaceutical potential. Each cone snail species that goes extinct takes its unique chemical toolkit with it, permanently, irrevocably. A 2003 paper in the journal Science warned explicitly about this possibility, arguing that the loss of cone snail species represents not just an ecological tragedy, but a medical one. We may be losing cures for diseases we have not yet learned to treat. The geography cone snail has survived for millions of years. It has weathered ice ages and warm periods, continental drift and sea level changes, mass extinctions, and ecological upheavals. It has evolved one of the most remarkable predatory systems in the animal kingdom, a biological weapon that combines ballistic precision, chemical sophistication, and metabolic sabotage in a package smaller than a human hand. It has, without knowing it, provided humanity with a powerful non-addictive painkiller, and may yet provide treatments for diabetes, epilepsy, and diseases we have not yet imagined. But survival in the 21st century requires something that evolution alone cannot provide. It requires that we, the species most responsible for the changes reshaping the world's oceans, choose to protect the habitats these animals depend on. It requires that we recognize that a slow-moving snail on a tropical reef is not just a pretty shell and not just a dangerous animal, but a living repository of biochemical knowledge that took 55 million years to accumulate, and that, once lost, can never be rebuilt. Tonight, somewhere on a warm reef in the Indo-Pacific, a geography cone snail is stirring from the sand. Its siphon is testing the water. Its harpoon is loaded. Its venom is ready. It has been doing this, night after night, for longer than our entire species has existed. It does not know that it carries inside its body the raw materials for a revolution in human medicine. It does not know that its habitat is shrinking. It does not know that its future depends on us. It knows only what it has always known: the chemical trace of a sleeping fish, the slow approach through the dark water, and the moment of the strike. Let us hope it keeps doing it for a very long time. If you enjoyed this deep dive into the world of the geography cone snail, take a moment to like, subscribe, and share this video. There are hundreds of extraordinary creatures in our oceans whose stories deserve to be told, and every subscriber helps us continue exploring them. Sweet dreams, and we will see you on the next dive.